Bismillahirrahmanirrahim. Assalamu alaikum. It's me, Abdul Basit, PhD scholar from the Department of Microbiology, Kohat University of Science and Technology, Kohat, supervised by the esteemed professor, Dr. Mubashir Hussain. The topic of my research project is in genetic diversity of multi-drug resistance Streptococcus pyogenes genes isolated from Tertiary Care Hospital of Khyber Pakhtunkhwa. The Streptococcus pyogenes bacteria, which is also known as group A Streptococcus, is a major pathogen which causes different suppurative and non-suppurative infection, starting from the superficial skin infections, cellulitis, etc., and the tonsillitis, acute sore throats, and leading to the glomerulonephritis. About 18 million streptococcus sore throat tonsillitis are reported yearly around the world, and around 5,18,000 patients with serious and complicated streptococcus pyogenes infection die yearly. Group A streptococcus induced sepsis in Pakistan are higher in the age of more than 15 years, and also causes fatal sepsis in pregnant women. Different factors which include increasing age, comorbidities, hospital stays, socioeconomic status affect the incidence of gas, particularly in the developing countries. A wide range of resistant mechanism is responsible for regulating a wide range of epidemic strains of group A streptococcus. But there are only a few reports in the Pakistani literature about drug resistance against erythromycin. The pathogenicity of the gas depends on the presence of different valent factors like M protein, F protein, and lipoic acid, which are encoded by specific virulence genes, also known as superantigens genes, which includes the following genes. The M gene, which encodes the M protein, is the main pathogenic factor of gas. The different types of M proteins are responsible for the pathogenicity in different strains. Despite Streptococcus pyogenes is of significant clinical importance in Pakistan, but minimal information exists from different parts of Pakistan, especially in Khyber Pakhtunkhwa. Most of work done previously is only in phenotypic antibiotic resistant profiles. Molecular typing of genes responsible for virulence and antimicrobial resistance for in-depth studying of gas is a need of the time. Here are the objectives of my research project: to characterize Streptococcus pyogenes based on M typing in clinical samples obtained from different tertiary care hospitals of Khyber Pakhtunkhwa, to study antibiogram profiles and MIC of antibiotic against Streptococcus pyogenes isolates, to determine the molecular basis of S pyogenes, antibiotic resistance, and virulence genes, to analyze the prevalence of Streptococcus pyogenes infections in KP and the associated risk factors. Ethical approval was obtained from the Custody Ethical Committee. Different samples, which included throat swabs, fluid samples, blood samples, were collected from different hospitals of Khyber Pakhtunkhwa. A pre-designed questionnaire was used to obtain the socio-demographic data, the socio-economic status of the patient, age, gender, profession, BMI, signs and symptoms, presence of comorbidities, pregnancy, diabetes, mellitus presence and absence, or antibiotic usage. This is the overview of our methodology. All samples were brought to the microbiology lab, Kohat University of Science and Technology. The culture was performed. The negative samples were excluded. The negative or the false positive samples were excluded. The positive samples were furtherly confirmed by using 16S RNA PCR, the phenotypicization, and risk factor analysis. The drug susceptibility testing was performed for all the positive targeted samples, which results in multi-drug resistance or XDR, extended drug resistance, which were the targeted samples of our research project. Furtherly, different PCRs were run for the M typing, for the antibiotic resistance, for different valence genes, and then these samples were sent for the gene sequencing. to the Brazil, and then phylogenetic and mutational analysis were done from the sequences data using bioinformatic tools. Different primers were used in the present studies. The PCR sampling conditions are as follows. Different using specific primers were used for the macrolide resistant gene analysis, which are for the presence of ARM A, B, and MFA. The following are the conditions for the PCR. M-typing analysis was performed according to the protocol of CDC. The 5' end of the DNA sequences was compared to the sequences listed in the CDC database for the comparison. The result, different samples, as we stated, throat swab samples, skin swab, or abscess swabs, urine, CSF were obtained. The total number of 500 samples were received from different tertiary care hospitals, which include HMC, KTH, LRH, MMC, ATH, KGN, and MMPH. The maximum number of samples were received or collected from the throats, followed by the abscess or skin samples, followed by the CSF, and very minimum number of urine were collected. Okay, the total 106 positive isolates were obtained, resistant isolates or the targeted isolates were collected from the 500 samples. Remaining samples which gave the growth of other bacteria excluded from study or no growth. The sore throat positive samples in 106, 48% samples were from sore throat, 22% samples were from sepsis, 18% sample were for cellulitis, and meningitis was 12% samples. This is a patient with septic sore throat. We can see that the tonsillitis infection. This is a beta hemolytic growth on streptococci genes on blood agar plate. You can see the hemolytic zone around the colonies grown on the blood agar. Different antibiotics were used, including the penicillin and the macrolide, ceftriaxone, or ceprofloxacin, and tetracycline. As we can see that penicillin and penicillin showed no resistance against the streptococci genes, but the streptococci genes isolates showed highest number for macrolides, tetracycline, and ceftriaxone. This is the determination of MIC. Azithromycin, clarithromycin, and ceprofloxacin samples were used in different concentration, which were compared with the Clinical Laboratory Standard Institute breakpoint. This is the molecular analysis of M-types, 960 base pairs. Molecular analysis of M-type revealed the band size of 960 base pairs. In clinical isolate, the positive control was used, which was Streptococcus pyogenes isolate characterized in the Department of Microbiology cohort, University of Science and Technology, and the negative control was also used. This is the distribution of different M-types in clinical isolates. As we can see that the highest number of isolate showed the M-type 42, followed by 27 samples which showed M-type 68. Prevalence of M-type based on gender and age groups. The maximum number of M-types were present in the male, and in the age group, maximum number of M-types were present in the 1 to 10 age group. The 72 isolates of that showed 84.6 had the ARMB gene, which is macrolide resistant isolates, and 14 isolates were 15.4% had the ARMA gene. This is the phylogenetic analysis of our isolates. As it was and it shows, or the phylogenetic analysis were done on the basis of amino acid slide differences. The SP006 is our isolates, and the second last Streptococcus pyogenes is the Streptococcus pyogenes database isolate. The A filter an Enterococcus outgroup was also used for spec genes. The conclusion recommendation showed that rate of microlytic resistance of streptococcus is currently very high in tertiary care hospitals of KP. And the resistant rate of macrolide was 86% for macrolide, followed by cephalosporin, fluoroquinolones, and tetracycline. MIC analysis revealed high level of resistance against clarithromycin and azithromycin. Molecular study revealed 13 different M types. This would provide new insights on the genetic diversity of the Streptococcus pyogenes in KP. Further studies are required to determine the molecular epidemiology of Streptococcus pyogenes on mass level. These are differences. Thank you.